Hey there. Well, welcome to the first AP Biology lecture that I've recorded so far. Well, I haven't recorded it yet, I'm doing that right now. Hopefully you guys can see all right and hear me all right. Again, I'm sorry that I'm such a novice at all of this stuff, but hopefully it'll get better. My apologies to Brendan, since you actually know things about making videos. This might be hard for you to watch. We'll see. All right, so we just finished talking about, in class the other day, if you were paying attention, Mendel. Remember Gregor Mendel? He was the monk who kind of is the father of modern genetics. And we talked about some of the experiments he did. And when we finished talking, we were talking about how he kind of discovered the law of segregation. Which sounds like a much worse thing than it is. It's nothing having to do with segregating people. This is about segregating genes and how genes are segregated as these kind of separate units. They don't blend and turn into other things. The characters themselves might be a blend, as we'll look at later, but the genes themselves do not blend. They stay the same. They are segregated from each other. Okay, so we were talking about that. Now, we're going to talk about some other things that Mendel learned from his experience, and then we'll talk about things that Mendel didn't learn from his experiments, but that we learned later on. So we talked about how he came up with that 3-to-1 ratio of different colors when he was looking at the colors of those different pea flowers, and he came up with kind of a bit of a model to explain this, and there are 4 different concepts that kind of work together to help explain that 3-to-1 ratio. And they're related to what we actually know now about how genes work. So first, alternative versions of genes account for variations in inherited characteristics. So there are multiple versions of those genes. We already used the word alleles in class the other day, but that's what we call those alternate versions. So for the pea flower, there's one gene, or one allele of the color gene that's purple, and one allele that's white. And then each gene is going to be at a very specific locus, specific location, on a specific chromosome. So the gene for color is going to be the same at the same place on the same chromosome in any pea plant. And the different alleles are just different versions that exist of that gene in that specific locus on that specific chromosome. So here, if we're looking at the gene for color in pea plants, color of flower in pea plants, we can see that the difference in the two alleles of that gene is just one base pair. So in the dominant gene, which is the dominant allele, the allele for purple flowers, you have a CG here. That's the base pair that you have there. And then in the allele for white flowers, the recessive allele, you have an AT base pair. That's the only difference in that whole gene. So that small, tiny little difference, that small mutation, essentially means that that gene, which happens to encode for an enzyme that helps to synthesize that purple pigment, makes it so that that gene can't be created, or that enzyme can't be created. So either that gene isn't being transcribed because of that, or the gene is being transcribed and translated, but it creates a non-functioning enzyme. One of those two, but all it is just one little base pair that's changed, and all of a sudden that enzyme either doesn't exist or doesn't work, and so that plant can't create those purple pigments, and so you don't get purple flowers, you just get white flowers. So that's just a very small difference, but that's the difference between the allele for purple flowers and the allele for white flowers. Okay, secondly, for each character, an organism is going to get two different alleles. One allele from each parent, one allele from the father, and one from the mother, of course. With plants, it's a little bit more complicated than that. And so he made this deduction without even knowing about chromosomes. He didn't know anything about chromosomes, but he was still able to make this deduction because of what he saw in his experiments. And we'll talk about how things get a little bit more complicated than that later. But, and he also said that the two alleles, well, he didn't say this. We know now that the two alleles at a particular locus, so the two alleles, two different versions of one gene, they might be identical, as in with the true breeding flowers that he was using, or they might be different. And we'll talk about the words for those in just a little bit. And we saw them having different ones in the F1 hybrids that we looked at last time. Thirdly, if the two alleles at a locus are different, then one of them, which we refer to as the dominant allele, determines what the organism's appearance is, and the other one, the recessive allele, has no noticeable effect on the appearance of the organism. And remember, dominant and recessive are words that Mendel was using at the time. So in flower color, any plant that has at least one allele for purple is going to be purple. It's going to have purple flowers. It has to have both of those alleles being the white allele in order for it to have white flowers. Fourth is the law of segregation. So two alleles for a heritable character separate or segregate during gamete formation. They are split up into two different gametes and then end up in different gametes. So an egg or a sperm is only going to get one of those alleles, and the segregation of alleles is going to correspond with the distribution of homologous chromosomes to different gametes in meiosis. Again, Mendel didn't know anything about chromosomes, so he wouldn't have been able to make that kind of statement. But we know now that those different alleles are going to separate or segregate into different gametes, and when they fuse together, they don't become one different allele, or one doesn't delete the other. You still have separate, segregated alleles. It's just that they, one of them is dominant over the other, or we'll look at some other cases. So that model accounts for that 3-to-1 ratio that he got in the F2 generation. And we can determine what the possible outcomes of different crosses are by using what's called a Punnett square. This Punnett square was named after a guy, I'm trying to remember his first name, a guy named Punnett, obviously. And he was a biologist, I believe he was a British geneticist. And he was going through grad school right around the turn of the century. And remember that Mendel's papers were kind of forgotten about up until 1900, when they were kind of rediscovered and people were more ready and willing to accept those ideas. So Punnett was going through grad school right at that time. And previously he had been unaware of Mendel's stuff, but then he got introduced to Mendel's stuff and started using or studying Mendelian genetics, which is what we refer to that style of genetics as. And he developed this way of determining the possible outcomes for the offspring. So, when we're looking at these, a capital letter is going to represent the dominant trait, and a lowercase letter is going to represent a recessive trait. So let's look at this. So if we were to take two different pea plants, and these ones, again, you have big P, little p, little p. So these are going to be true breeding peas. And if you were to take those and cross them by each other, this one only has 2 dominant alleles to give. This one only has 2 recessive alleles to give. So you know that all of the flowers that you're going to get, they'll have one from this flower, one from this parent, and one from this parent. So all of the offspring that you're going to get are going to have one of each of those alleles. They will all be, they'll all be purple because they all have at least one purple allele. But if you were to cross that with itself, you could use this Punnett square to determine what kind of ratios you might get of offspring. So you just put the 2 different alleles from one parent on this side. So this top one is the sperm from the F1 plant. And then you have eggs from the F1 plant. And each of those is giving 1 purple allele and 1 white allele. 1 purple allele, 1 white allele. And then you just kind of say, okay, this is going to have 1 big P, 1 big P, this is going to have 1 big P and 1 little p, this is going to have 1 big P and 1 little p, and these will both have 2 little p's. And so once you write those down, you see, okay, you have 1 with 2 dominant alleles, 2 of them with 1 of each, and 1 with 2 recessive alleles. And that shows that you would have that 3-to-1 relationship, that 3-to-1 ratio. So I was trying not to use these words until we got to this slide, but 2 really important words to use when you're describing the genetic makeup of an organism in reference to a specific character are homozygous and heterozygous. Homozygous means that they have 2 identical alleles. So in the case that we were just looking at, that would be 2 purple alleles or 2 white alleles. And then heterozygous is when you have 1 of each. So that would be in that Punnett square that we were looking at, those 2 that had 1 purple allele and 1 white allele, those are both heterozygous. And so you can be homozygous dominant, as with the purple flower, or homozygous recessive, as with the white flower, and then heterozygous is just 1 of each. So homozygotes are what we refer to as true breeding, or what Mendel would have referred to as true breeding. Heterozygotes are not true breeding. Okay, so because of the different effects that alleles can have, whether they're dominant or recessive, organisms don't necessarily show what their genetic makeup is. And in order to kind of distinguish between their genetic makeup and what they actually look like, we use the words phenotype and genotype. So phenotype is what the organism looks like. And it's not always looks. Sometimes a phenotype is behavior. Sometimes a phenotype is the internal anatomy. But in the case of all the traits that Mendel was looking at, of course he wasn't looking at the internal anatomy of all these plants, it's going to be the physical appearance. So the phenotype is kind of the outcome, and the genotype is what's in the blueprints, what's in the DNA. So the genotype of those flowers would be big P, or big P, little p. But the phenotype of all those would be purple. Yeah, like it says right there. So here you go. So you can have either a homozygous dominant or heterozygous plant that's going to have purple flowers. In that case, the phenotype is purple flowers, the genotype is homozygous dominant, heterozygous. And then you can also have, of course, white, but only with the homozygous recessive genotype. So, because, as we saw in the last slide, both the homozygous dominant and the heterozygous genotypes will display the same phenotype, you can't know for sure whether or not, say, a plant with purple flowers is homozygous dominant or heterozygous. And so we have to have a way of kind of determining that. So if you're doing something like breeding plants, you can do what's called a test cross. And in a test cross, you can take whatever that mystery individual is, some plant with purple flowers, and breed it with a homozygous recessive individual. And again, you can know if an organism is homozygous recessive because that's the only genotype that will lead to the recessive phenotype. So, if you were to take a plant with purple flowers of unknown genotype and breed it with a plant with white flowers, you could tell whether or not that plant was heterozygous or homozygous dominant. Because if any of those offspring display the recessive characteristics, that means that the mystery individual must have been heterozygous. Otherwise, it would only be able to donate the dominant allele, which would mean that all of the offspring would show the dominant phenotype. All right. So this is a visual of what that would look like. If you had a homozygous dominant individual, this is what you would see. All purple flowers, because this homozygous recessive individual will be giving only recessive alleles, but this one would only be giving dominant alleles, so all of them would turn out heterozygous, and you have all purple flowers. If, on the other hand, you had a heterozygous organism, then its gametes would have half of its gametes would have the dominant allele, half of them would have the recessive allele, and you would end up expecting to see about 50-50 with 2 heterozygous individuals and 2 homozygous recessive individuals. Alright, hopefully that makes sense. So, the law of independent assortment. We already talked about independent assortment in the last chapter, I think the end of the last chapter. But this is another conclusion that Mendel kind of came to. So he came to that law of segregation by following just a single character and tracing that. So we were looking, for example, at the flower cup. And the F1 offsprings that he produced in those crosses were known as monohybrids because they were heterozygous for just one character. And then when you cross those together, you get what's called a monohybrid cross. But he had to come across the second law of inheritance by following 2 characters, which makes things a little bit more complicated. So he had to cross 2 true breeding parents for 2 different characters and produce dihybrids because it was 2 different characters. And those were heterozygous for both of those characters. So a dihybrid, and of course, if they're heterozygous for both of those characters, that means that the phenotype that they're going to be showing is the dominant phenotype. So a dihybrid cross, which is a cross between 2 dihybrid F1 generations, can determine whether or not those 2 characters are going to be transmitted to offspring as a package or independently. And if you remember from our discussion about independent assortment, we know that chromosomes don't line up along that metaphase plate, or the homologous pairs of chromosomes don't line up along that metaphase plate with all of the paternal chromosomes on one side and all of the maternal chromosomes on the other side. They just assort themselves independent of where the other ones are. And that is something that he was able to determine. Because if you had all of those chromosomes lining up, and of course again, Mendel wasn't thinking in terms of chromosomes, but But those characters obviously weren't linked to each other, because if they were linked to each other, if, for example, here we have two characters that he's looking at, yellow versus green, and round versus wrinkled. So the big Y is yellow, little y is green, big R is round, little r is wrinkled. So he crossed one parent that had homozygous dominant and the other for both traits, and the other that was homozygous recessive for both traits, got a dihybrid cross, big Y, little y, big R, little r, and then bred that with itself. And so if those two were linked, you would expect to only see the round yellow peas and the wrinkled green peas. But that's not what he saw. He saw a mix. He saw some wrinkled and yellow ones, some round and green ones, and then the round yellow and the wrinkled green ones. So that shows that those two were not linked to each other. And the fact that that was the case is another instance, like we talked about in class, where Mendel got a little bit lucky in the characteristics that he was looking at. And we'll talk about that in just a second. So the ratio for this one was 9 to 3 to 3 to 1. You can see you have 9 of these yellow round ones. Both of those are dominant traits. And so the individuals that are heterozygous for either of those alleles are going to be showing the dominant trait. And you got 3 of these yellow wrinkled ones, 3 of the green round ones, and 1 of the green wrinkled ones. So, yeah, again, 9 to 3 to 3 to 1 is the ratio. And that's not, these ratios that we talk about, they aren't exact, but that's about the ratio that you would expect. And then when you draw out one of these Punnett squares, you can see that what the ratio you would expect would be. Okay, so using that dihybrid cross, he was able to come up with the law of independent assortment, which states that each pair of alleles is going to segregate independently of each other pair of alleles during gamete formation. So not only are those alleles themselves, like the two different alleles for the same gene, segregating, but they're doing so independently of the alleles for other genes. And so the catch there is that this law only really applies to genes that are not on the same chromosome, or ones that are far apart. Because, let's draw this out really fast, you remember that we have crossing over happening, right? So if we had a chromosome made up of 2 sister chromatids, if you have genes that are very far apart from each other on the same chromosome, I hope you guys can see this. Can you see this here? It might not be dark enough. Oops. If you had genes that were far enough apart from each other, say here and here, then there's a chance that one of those, that crossing over will occur, and those might not end up in the same gamete. But if they're closer to each other, or if they are nearer to the centromere there, then it's very unlikely they're actually going to separate. If they're on completely different chromosomes, they will separate, or it's much more likely that they will separate. But if they're too close together, or if they're too close to the centromere on the same chromosome, then they're not likely to separate. And so if you would have been studying 2 different characters whose genes were close to each other on the same chromosome, he probably wouldn't have gotten the same results, and probably wouldn't have come up with this law of independent assortment. So, you got lucky there. All right, let's see here. Probability laws. I might cut here and then come back and do this in just a little bit. So I'll be back in just a second. All right, let's get through this probability law stuff. I need to go get lunch soon. All right, so probability laws. So these probability laws are part of what govern Mendelian inheritance. So Mendel's laws of both segregation and independent assortment reflect these rules of probability. So it's like when you're tossing a coin. When you're tossing a coin, the outcome of each coin flip doesn't really have any impact on the outcome of the next toss. So if I was to flip a coin and get heads, you might say it's less likely that I'm going to get heads the next time, but really it's still just a 50-50 chance. Now the probability of me flipping a coin 10 times and getting heads every time is pretty low, but if I were to flip it heads 9 times, the 10th time is still a 50-50 chance. So similarly, the way that alleles of one gene segregate from each other is independent of the alleles of other genes. And we can use the multiplication rule to kind of calculate some of these different characteristics. And so the multiplication rule states that, this is a little bit wordy, the probability that 2 or more independent events will occur together is the product of their individual probabilities. So the probability that I'm going to flip a coin 10 times and get heads every time is the product of the individual possibilities. So in that case it would be 1/2 times 1/2 times 1/2 times 1/2 times 10 times. So probability in an F1 monohybrid cross can be determined using this multiplication rule. Each gamete, it's just like flipping a coin, each gamete has 1/2 chance of carrying the dominant allele and a 1/2 chance of carrying the recessive allele. So, if we're looking, say, at the round characteristic, which they have the alleles here for round and wrinkle, but then they're showing pennies. I don't really know. So let's say that heads is dominant, tails is recessive. Well, if we were to flip it twice, right? If we were to flip this coin twice, once for each allele, there's half of a chance, or a 50-50 chance that we're going to get dominant or recessive, heads or tails. So the chance of us flipping it twice and getting heads both times is 1 out of 4. The chance of us flipping it twice and getting 1 heads, 1 tails is 1 out of 4 as well, to get heads the first time and tails the second time, and 1 out of 4 to get tails the first time, heads the second time. And then the likelihood of us getting tails both times is also 1 out of 4. So it's 1 out of 4 for each of those outcomes. But if we were to look at just the chances of us getting one of each, it's actually 1/4 plus 1/4 to make 1/2. So the addition rule states the probability of any one of 2 or more exclusive events will occur is calculated by adding together their individual probabilities. That's what I just did. So the rule of addition can be used to figure out the probability that an F2 plant from a monohybrid cross will be heterozygous rather than homozygous. Let's look at an example of that. Never mind, we don't have an example of that coming up. There is an example coming up. But, so you can use that to figure out what the different possibilities are. Or what like with heterozygous versus homozygous. There's a 1/2 chance, which you get from adding 1/4 plus 1/4, and you would get 2 over 8, which is 1 over, no, that's not true. You get 2 over 4, sorry. You get 2 over 4, which would then be 1 over 2. Okay. So we can apply these different rules of probability in order to predict the outcome of kind of more complicated crosses. So we call these multi-character crosses. Multi-character crosses are the equivalent to kind of to 2 or more independent monohybrid crosses occurring at the exact same time because of that independent assortment thing. Because those different genes, as long as they're not on the same chromosome too close, are independently assorted, we can look at these as different monohybrid crosses happening at the same time. So, for example, if we were looking at a cross between, is this working? Okay. If we were looking at a cross, come on, there we go, between Y, Y, R, R, oopsies, that should be a lowercase, R, R. If we had 2 heterozygous individuals like this, the chances of getting Y, Y are 1/4, and big Y is 1/4, and the chances of getting big R is 1/4. So the likelihood of having both of those, which is homozygous dominant, is 1/16. And the likelihood of getting a heterozygous plus homozygous dominant is going to be 1/8, because the likelihood of getting a heterozygous YY is 1/2. The likelihood of a homozygous RR is 1/8. So let's show that here. So if we had big Y, little y, big Y, little y, you can see that we would have big Y, big Y, big Y, little y, big Y, little y, and little y, little y. So there you go. 1/2 to get heterozygous, 1/4 to get homozygous. Alright, hopefully that makes sense to you. One disadvantage of doing this by video is that I can't actually ask you guys if this is making sense. But if any of this isn't making sense, just write down your questions or remember your questions or whatever, and bring them into class the next day that we have class. I was going to say tomorrow, but that might not be tomorrow. And bring them in, and we'll talk about them in class. Okay, so this is a more complicated one. Let's see, this is the likelihood of getting, okay, so this is if you were to cross a completely heterozygous individual, PP, Y, Y, R, R, by heterozygous, and then homozygous recessive. Wait. Yes. Let's see. Y, Y, R, R. Okay. So if you were to cross these 2 individuals, let's look at the probabilities of these different things. So the probability in this case of getting, let's see, so we have P, 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 P. I'm not actually going to write out all of the little ones in there, but I'll just draw it out because I think that'll still make it a little bit easier to visualize. And big R, little r, little r, little r, boop, 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 bam, bam. Okay. So let's see here. Probability of getting little p, is only 1/4. That's only going to be this square, right? So that's 1/4 there. 1/2 to get little y, because both of these are going to be little y. And 1/2 to get big R, little r, because that'll be both of these ones. So you can calculate all these possibilities of getting these combinations just by multiplying these by each other, right? So because we can look at these as 3 different monohybrid crosses happening at the same time. That means that we can multiply the probabilities of each of these individual characters in order to figure out the probability of any combination of those characters. So the probability of getting this combination, little b, big little y, big R, little r, is going to be 1/4 times 1/2 times 1/2, or 1 out of 4 times 1 out of 2 times 1 out of 2. So that'll be 1/16. And then if you are trying to calculate the chances of getting at least 2 recessive traits, you can add up all of these, because all of these have 2 different recessive traits. At least 2. This one has these 2 recessive traits. This one has these 2 recessive traits. This one has these 2 recessive traits. This one also has these 2 recessive traits. This has all 3 recessive traits. So you just add up all the possibilities of getting at least 2 recessive traits, and then using that addition rule, you can get the possibility of that outcome. Okay, these are a little bit complicated, but we will be doing some kind of homeworky type problems with them, and that'll give you some practice, which will hopefully make things a little bit easier, because this is kind of a mathematical thing, statistical thing. All right, so let's go beyond the Mendelian genetics. I hope this isn't going to be too long of a lecture. I apologize if it is. Oops. We need to get through steps though, so I'm not really that sorry. Okay, so genetic stuff gets a lot more complicated than just kind of the Mendelian genetics that we've been looking at so far. And most of the time, those genetics, the relationship between genotype and phenotype, aren't as simple as what Mendel saw when he was doing his experiments. A lot of heritable characteristics are determined by multiple genes or multiple alleles of a certain gene. The basic principles, though, that he discovered were... there's something going on. I should probably go investigate that. I'll be right back. Okay, sorry about that. There was noise at the door and I decided to see what was going on. Okay, so, where was I? Well, we'll just start this over again. So, inheritance patterns, or genetic stuff, is a lot of times a lot more complex than what Mendel kind of discovered. So let's go beyond those basic Mendelian genetics and look at some more complicated stuff that he wasn't really able to determine by his fairly simple experiments. A lot of times the relationship between a genotype and a phenotype is not going to be as simple as what Mendel discovered. In fact, kind of most of the time it's going to be more complicated. And so like I've said a couple times now, Mendel was maybe lucky, or maybe he just chose well the characters that he was looking at. A lot of heritable characters or traits are not just determined by a single gene or by 2 alleles, and we're going to look at some of the examples of those things. But the same principles that he discovered still apply to these different more complex genetic stuff, these genetic things, but he probably wouldn't have discovered those principles if he was studying something more complicated, because he wouldn't have observed the things that he observed. Okay, so let's see here. So these are some of the situations in which you're going to see deviation from those simple Mendelian genetic principles. If you have alleles that are not completely dominant or recessive, we'll see that. If a gene has more than just 2 alleles, we'll see some examples of that. And when a gene produces multiple phenotypes, and we'll also see that. So not always, when you're looking at genetics, is an allele completely dominant or completely recessive. There are varying degrees. And there are kind of three different types of dominance relationships that we're going to look at. There's complete dominance, which is what we've seen so far, and that's when there is just dominant and recessive. And if there's a heterozygote, they're going to be showing the dominant trait. But there's also incomplete dominance. And in incomplete dominance, you have the heterozygotes showing kind of a mix between the dominant trait and the recessive trait. And then there's codominance. And in codominance, you have two dominant alleles affecting the phenotype in separate, distinguishable ways. So they're both kind of showing their own characters. And we'll see an example of that as well. All right, so let's look at some snapdragon flowers in order to take a look at incomplete dominance. So if we were to do a similar thing to what Mendel did with his pea plants, we would take two true breeding flowers, so one true breeding flower with one allele, and another true breeding flower of the other allele. And we would cross those true breeding plants. And in the F1 generation, like we would expect from Mendelian genetics, we would see heterozygotes. And this is what we see. We see heterozygotes in this generation, all heterozygotes, because there's only one allele coming from this side and only one allele coming from this side, so every single individual, every single offspring of that next generation is going to have a mix of both. But, unlike with what Mendel saw in his experiments, in these snapdragons, you don't see just a dominant allele coming through in the phenotype. You see a mixture. So, in this case, you have red and white making pink. Crazy. And then, when you cross these heterozygotes with each other, you're going to get, again, a mix of homozygous, heterozygous, and or homozygous dominant, heterozygous, and homozygous recessive. But instead of seeing that 3-to-1 ratio that Mendel saw, you're going to see a 1-to-2-to-1 ratio, because you're going to have one homozygous dominant, or I guess just homozygous, one of one type of homozygous, two heterozygous, and then another homozygous. Interesting. So you can use these ratios to figure out what the dominance relationship is between different characters, or different alleles. All right, so the dominant allele doesn't always, they say here, subdue, which is kind of a funny image, doesn't always subdue a recessive allele. That's just not always how they do their thing. Sometimes they, well, anyways, that's just not what's actually happening at the microscopic level. All they are is just different variations in the nucleotide sequence, because all a gene is just a piece of DNA. And so, like what we saw when we were looking at the gene for flower color in peas, if you'll recall, it was just one little base pair that was changed. And so that's all it is a different sequence of nucleotides. So for any character, the whole dominance and recessiveness is going to depend on how close we're looking. For example, there's a disease called Tay-Sachs disease, and in Tay-Sachs disease, which I'm not 100% sure I'm pronouncing that correctly, in Tay-Sachs disease, if a child is born with Tay-Sachs disease, they will die after a few years. And that happens because it creates a dysfunctional enzyme, and that enzyme is kind of in charge of breaking down lipids in brain cells. And when that enzyme isn't able to do its job, then you have an accumulation of lipids in those brain cells, and eventually there's seizures and a whole bunch of bad stuff, and the child will eventually die. Which is very sad. But if you look at the organismal level, the allele is recessive. So that means that at the organismal level, when you're looking at just whether or not an organism has the disease, you'll say, oh, well, that's going to be a recessive allele because you have to have 2 of that allele in order to have the disease. If you're heterozygous, you will not have the disease. You have to be homozygous recessive. So if you were looking at whether or not an organism has a disease and you made a Punnett square, you would see a 3-to-1 ratio. But at the biochemical level, the phenotype is incompletely dominant. Because at the biochemical level, if you actually look at what's happening in their brain, they do not, the individuals with Tay-Sachs disease, or I should say individuals that are heterozygous for Tay-Sachs disease, so they don't have Tay-Sachs disease, but they have one of each allele, those individuals are not going to be creating, or yeah, they have lower enzyme activity, if you're looking at the biochemistry of their brain. There's lower enzyme activity than you would see in a homozygous dominant individual. So an individual that's homozygous for the normal allele would have higher enzyme activity in their brains than that heterozygous individual. So you would say that's incompletely dominant because individuals who are homozygous for the normal allele have high activity of the enzyme that's breaking down those lipids. And individuals who are heterozygous have, oh, I'm getting a phone call. I'll not worry about that for now. Individuals that are heterozygous are going to have slightly lower enzyme activity. And then individuals who are homozygous recessive have no enzyme activity. And those lipids build up to the point that the individual dies. But since having only half of it, roughly half of the enzyme activity of a normal individual is evidently enough to keep those lipid levels down sufficiently, then they don't show up as having the disease. So again, looking at the organism, they appear to be a normal, normal individual without any disease. But then at the molecular level, the alleles would appear to be codominant because, again, in codominance, you see both of those possible characters coming through. And what you see, if you look at the molecular level, is they are the individuals who are heterozygous for this gene actually are producing good enzymes and messed up enzymes. And so they're producing functional and dysfunctional enzymes. So both of those traits are being displayed, and if you look closely enough, you would say that looks like a codominant relationship between those two alleles. So it depends on which level you look at. All right. Another thing that can be kind of confusing is that dominant alleles are not always more frequent than recessive alleles. The words dominant and recessive simply refer to the relationship between those two alleles, and they say nothing about the frequency of those alleles. For example, it's actually, I would think quite a few, because there are quite a few babies born in the United States every year. But 1 out of 400 babies born in the United States is born with extra digits, extra fingers or toes. And that is thought to be a dominant allele. So that means that if you are heterozygous or homozygous dominant, you're going to have multiple fingers. But it's still rare. It's still only 1 out of 400. So just because it's a dominant allele doesn't mean that it's going to be a more common thing. Now, if at some point in human history, for some reason, people found people with extra digits more attractive, and they got more breeding opportunities, then you might see an increase in that allele. But that wasn't the case, and so the frequency of alleles is still much lower than the frequency of the recessive allele, which gives us just 10 fingers and 10 toes. Anyways, so that's kind of interesting, and that's kind of a tie-in as well to what we were talking about the other day of the changes in frequency of alleles and natural selection and kind of how that happens. Because again, if even, I don't know if you can really call it natural selection if it's humans finding something more attractive. But if humans were to find more than 10 fingers or 10 toes attractive, then it could have been that dominant allele would have been selected for and would have become more common, and you would have seen more children being born with multiple or more than 10 fingers or more than 10 toes. So anyways, that all has to do with the frequency of alleles, which doesn't affect dominance or recessiveness of an allele. Okay. So multiple alleles is another thing that kind of makes things more complex. Most genes in populations are going to have two allelic forms, but if another mutation arises, then there might be another allele that is introduced. And so let's see. For example, there are 4 different phenotypes with the ABO blood group, and that's why. Well, yeah, we'll talk about that in just a little bit. So the ABO blood group, some of you might know your blood type. I don't actually know mine, but we're not going to be talking about the whole positive negative thing. But there are 4 different blood types that come about from 3 different alleles. There are 3 alleles for the enzyme that attaches A or B carbohydrates to red blood cells. So these are carbohydrates that are going to be sticking out on the outside of your blood cells. And these enzymes are going to be encoded by either the IA allele, so the enzyme that is created by that allele puts the A carbohydrate on the outside of the cell. The ones for the IB allele has the B carbohydrate, and the ones for the little I allele doesn't do anything, doesn't add either. So, here are the 3 alleles, here are the carbohydrates that they put on the outside of the blood cell. So, if you have the genotype of IA, homozygous, or IA and then little I, then you're going to have A carbohydrates on the outside of your red blood cell, which gives you the A blood group. If, on the other hand, you have you're homozygous for IB or have IB and little I, then you're going to have the B carbohydrate on the outside of your blood cell, and you'll be blood group B. If you have both IA and IB, then you'll be AB. Or if you have both little I, then you become O because you don't have either of those carbohydrates on the outside. And remember, carbohydrates on the outside of cells are usually used for, anyone? I'm not actually going to wait until one of you answers. They're usually used for cell recognition. Okay, and that comes into play when you talk about why you can't give blood transfusions from someone with type A blood to someone who has type B blood. And we can talk more about blood types later. So anyways, you should be able to, is there another, no, this is the last one. You should be able to look at these and figure out what the dominance relationship is between these. And so that is the question that I would like you to bring to class. I think it should be Monday. Yeah, Monday. So on Monday, today's the 3rd, 4th, 5th, 7th. Monday the 7th, I would like you to come to class with an answer to the question, of what is the dominance relationship between the alleles of the ABO blood groups. Okay? Cool. And I will probably not repeat the question, I'll just say what's the answer to that question I asked, just to see if you were paying attention. So if you happen to be skipping through this at random intervals and stumble across this section, congratulations, you got lucky. All right. Okay, so another thing we're going to talk about is pleiotropy. And pleiotropy is when a single gene is going to have multiple phenotypic effects. So a single gene is going to affect more than just one character. There are pleiotropic alleles that are responsible for certain diseases like cystic fibrosis and sickle cell disease, and those diseases have multiple symptoms, but they're caused by just one gene. So that's one gene that's having multiple effects, in this case, causing multiple symptoms in different diseases. And then there's also multiple genes that can have effects on other things. And one condition where that happens, or I should say, one way that happens is by what we call epistasis. Epistasis is, I think, Latin. It means like standing on top of. And that's when one gene is going to alter the effect of another gene in a different locus or on a different chromosome. And one example of that is with Labradors. And in Labradors, like with several other types of mammals, the color of their coat, the color of their hair or fur depends on two different genes. So one gene is going to determine the pigment color, and then the other gene is going to determine whether or not that pigment is at all deposited onto the hair. So here is a Punnett square of Labradors, which are very adorable, Labradorable. And you can see that there are two different genes. There's big B, there's two genes, there's B and E. So big B allele is going to be the black hair allele. Little b is going to be the brown hair allele. And then big E is going to be depositing that pigment. So that means that there is color. And little e means no color. So these are completely dominant, which means that if you have big B, little b, you're still going to be black hair. And if you have big E, little e, there's still going to be color. So it's completely dominant, but anything that is recessive or is homozygous for the recessive allele, doesn't matter if it's little b, big B, or big B, they're all going to be, well, I say no color, but that turns into those golden retrievers, the white labs, I think is what they're usually referred to as. White labs. If they are either heterozygous or homozygous dominant for that color, whether or not there will be color gene, then they will have color. And if they are homozygous recessive for the black or brown color gene, then they will turn out to be chocolate labs. And if they're either heterozygous or homozygous dominant for the black hair, brown hair gene, then they're going to be black labs. So black labs, chocolate labs, and white labs. And that's how the genetics of those works. Interesting stuff. Then there's polygenic inheritance. So there are what are called quantitative characters, and quantitative characters are characters where there's like a kind of a spectrum of different phenotypes. And usually when you see that kind of spectrum, that means that there's this polygenic inheritance where you have multiple genes working on a phenotype, And skin color is thought to be an example of this type of polygenic inheritance. As far as I know, the exact mechanisms of like the genetic inheritance of skin color are not completely known, but this is probably actually a simplified version of that. It's probably more complex than this, but this is kind of a simplified example of how this might work. So in this example here, this is another Punnett square. It just looks a little bit different and it's a little bit more complicated. But there are 3 different genes. So you can see there's gene A, gene B, and gene C. These were named very originally because, again, this is somewhat hypothetical. And you can be dominant or recessive for those. And they are not completely dominant. So that means that if you are heterozygous for one of those genes, you're going to be a lighter skin color than if you're homozygous for one of those genes. So it's incomplete dominance, and you have 3 different genes. And so you get this kind of blending effect where if you are, let's see, if you have 0 dark skin alleles, you're going to be much, much lighter. And if you have all 6 dark skin alleles, then you're going to be very, very dark. And this kind of gives you the frequency of those. And so if you were to cross 2 individuals that were heterozygous for all 3 of those genes, you get a ratio of 1 to 6 to 15 to 20 to 15 to 6 to 1. So yeah, it's there's a spectrum here, a big spectrum of light to dark. And so that's thought to be kind of how skin color works. It's probably more than 3 genes, but just to keep things a little bit more simple, we're just going to look at that example. All right, so phenotype can also be affected not just by your genotype, but also by your environment, which makes things even more confusing. And so let's see. When phenotypes can be dependent on environmental factors as well as genetic factors, we call it multifactorial because there are multiple factors that are affecting it. One example of that is in hydrangeas, and this picture looks like it was tweaked with a bit to make it look a little bit more blue than it really is, but the example still works. So in these hydrangeas, these can be genetically exactly the same, but depending on the soil conditions, the soil acidity, you can get pink flowers or blue flowers. So from one environment to the other, you're going to have different phenotypes. And where are we? Okay, I'll finish this slide and then I'm going to call it quits because I'm starting to get hungry and I've been at this for a really long time and you've probably been watching this for a considerable amount of time. And I still have to edit this video. So we'll see how that goes. All right, a Mendelian view of heredity and variation. So an organism's phenotype is not just the physical appearance. So Mendel was looking at the color of the seeds, or the color of the flowers, or how wrinkled the seeds were. Those are all physical appearance things. But that's not the only way the phenotype expresses itself. Phenotype can also be internal anatomy, the actual physiology, how your body works, or behavior. And the organism's phenotype doesn't only reflect its genotype or you know, the genetic makeup of their parents. It also reflects the unique environmental history. So just like those 2 hydrangeas, they can be genetically identical, but the phenotype is showing not only their genetic makeup but also their environmental influences. Okay. I'm just going to call it quits there, and hopefully you made it all the way through. We'll see. Yeah. All right. I will see you guys in class.